start this.
All right, hi everybody. Welcome to week 6, review session 1. We're going to be going over cell signaling, receptors, and then some examples of those. Okay, so pretty much everything that the cell does, everything that goes on in cells is through signaling. So obviously that's pretty important to know. And there's 2 types of signals, as well as 2 types of receptors, but like we'll get into that later. So 2 types of signals. There's hydrophobic ones, hydrophilic ones. Here's some examples of each of those. Knowing the property of your signal can tell you about like the signal speed, the signal transduction. It's pretty important to bear in mind, you know. So I know this is pretty straightforward, but let's start off with a question. So let's say your cell needed to respond to some kind of stimulus very, very quickly. So would you expect that signaling molecule to be hydrophobic or hydrophilic? And again, as just a refresher, hydrophilic means it's polar. Hydrophobic means it is nonpolar. So what do you guys think? If a cell needs something to happen very, very quickly, would you expect the signal to be hydrophobic or hydrophilic? You could drop it in the chat. Like I said, I can't see the chat, but you could drop it there if you'd like. Sorry, did someone say something? Oh, I found the chat.
Yeah, I said it was hydrophilic because it's coming from an external source.
Okay. Does anybody have any other guesses? Can you elaborate as to what you mean about an external source?
Well, it's just like coming from the question. If the cell needed an immediate response from an external stimulus, I would assume that.
Yeah. Okay. I'm sorry. That was phrased incorrectly. I mean, if like let's say something was going on, something in the environment is going on. What kind of signal would you expect if the cell needed like a very quick response? So okay, I'm sorry. Maybe external stimulus is like kind of throwing it off. Let's say the cell just needed a very, very quick response to something. Any guesses? So for something like this, I'm sorry, like I probably worded it a little incorrectly. That was my bad. But if the cell needs something to be done very quickly, that signal should be hydrophobic. And it should be hydrophobic because that signal will be able to bypass the phospholipid bilayer very easily. And if the signal could bypass the bilayer without needing a receptor or anything, it could go straight through the cell membrane, straight into whatever protein it needs to find or whatever organelle it needs to find, whatever the deal is. But if you have a hydrophilic signal, that signal can't pass through the phospholipid bilayer because it's hydrophilic. So in that case, it would need to first find a receptor. That receptor would then need to communicate the information to another protein, in which that protein would have to talk to like other proteins or whatever. But if you have a hydrophobic signal, you skip all that step. You skip the need for a surface cell receptor and for that surface cell receptor to find another protein. Does that kind of make sense?
Yeah, as soon as you started explaining it, it just immediately came back from the lecture.
Yeah, no problem. And I understand, like, I kind of phrased that a little, I know external stimulus is worded a little weird, so sorry for that. But okay, any questions about that before we go? Sweet, okay. Okay, so here's an example of like real life cell signaling. So we have neutrophils in our body, which are like white blood cells. And let's say you have like an E. coli infection or whatever. How do your white blood cells know where to go, right? Like if you get a cut on your skin, like let's say you tripped and you scraped your knee, how do your white blood cells know that there's a possible infection there? Like how do they find that? And it's through cell signaling. So bacteria or other pathogens, they give off chemicals which can be interpreted as like a signal. Neutrophils, like other cells, are sensitive to those signals. So when they detect that bacteria or a virus or whatever is releasing these chemical signals, it will actually, those chemicals that the bacteria are releasing will find the neutrophil, the white blood cell. It'll bind to the white blood cell and it'll tell the white blood cell like, okay, this is where it's coming from. Like, this is where I need to move. So this is just one example of how cells respond to signals. This is also called chemotaxis, by the way. Chemo meaning chemical, taxis is movement. So yeah, the white blood cell is going to detect these signaling molecules from bacteria or a virus or whatever. It's going to take that signal and it's going to respond by propelling its flagella or whatever it has, in this case, the actin filaments. Okay, perfect. Yeah, it's just going to propel itself to wherever that signal came from. Another example is when there's this virus called Phytophthora, which actually started the Irish potato famine. And this fungus, sorry, it's not a virus, it's a fungus. This fungus has, or like it's made by these little spores, which also move by chemotaxis. When a plant starts growing, it releases chemicals into the soil, and this fungus detects those chemicals from the potato plant or from whatever plant it is. The pathogen detects those chemicals and it moves towards wherever that signal is coming from. And that's how it messes up your potatoes or your plants or tomatoes, things to that effect. Okay, so any question on that? Sweet. Okay, so I mentioned before that we have hydrophilic and hydrophobic signals, and this is just putting into context what I was talking about before. Hydrophobic signals, because they are hydrophobic, they can interact with the hydrophobic layer of the phospholipid bilayer, so they're able to diffuse pretty quickly. So I don't know if you can see my cursor, but down here, this is an example of signals like just directly diffusing straight through the phospholipid bilayer. Again, they're hydrophobic, just like the tails. So they're able to slip through easily. But if you have a hydrophilic receptor, a hydrophilic signal, that hydrophilic signal needs to find a receptor on the surface, and then that receptor needs to transduce whatever signal it needs to. So for that reason, it is slower than hydrophobic signals, because again, hydrophobic could go straight through, whereas hydrophilic, they need to find specific receptors, and those receptors need to do their own thing. Okay, any questions about that? Okay, so receptors, just like there are hydrophilic and hydrophobic signals, there are also hydrophilic and hydrophobic receptors. So I probably should have made this a question, but that's okay. Extracellular ones, or actually, yeah, I will phrase it as a question. So why do you think that extracellular receptors, like it says here on the slide, so extracellular receptors, they usually bind to hydrophilic ones. Why can't you have an extracellular receptor that could bind to hydrophobic signals? Or vice versa? Or why can't you have an intracellular receptor that binds to hydrophilic receptors? Do you guys have any ideas? Okay, so this goes back to the idea of being hydrophobic and hydrophilic. If a signal is hydrophobic, it is very easy to diffuse straight through the lipid bilayer, and it doesn't need to find a cell surface receptor. And mind you, a hydrophobic species interact with other hydrophobic species. Hydrophobic and hydrophilic, they do not attract each other. They repel each other. So it makes sense that the extracellular signals, the one outside of the cell that are on the surface of the cell, it makes sense that those are hydrophilic, because hydrophilic signals can't go through the lipid bilayer, so they need a hydrophilic receptor for a hydrophilic signal. And likewise, intracellular signals, so receptors inside the cell, those are typically hydrophobic, because again, hydrophobic things need to combine with other hydrophobic things. It makes sense that they're inside the cell, because hydrophobic signals can slip through the bilayer with no problem, and then they could find their hydrophobic receptor inside the cell. So does that make sense? Any questions about? Sweet. Any questions before I move on with this?
So bottom line is that the extracellular signals, since they are hydrophilic, they will have hydrophilic receptors, and vice versa for hydrophobic. Hydrophobic will have the receptors, but it's all intracellular because they can pass through the lipid bilayer.
Exactly. Exactly. Let's say you have a mutation of an intracellular receptor that can only attract hydrophilic signals. How well do you think that's going to go for the cell?
Could you repeat that one more time?
Yeah, let's say that there's a mutation in an intracellular receptor that changes it to where it only has an affinity for hydrophilic signals. What do you think, like, the outcome?
Well, either the receptor is going to remove to become a surface receptor so it can interact with those hydrophilic signals, or it's never going to have any interactions inside the cell because there's no hydrophilic molecules that can pass through that membrane.
Right. Okay. Yeah. I was thinking more about like the second part that you said. That intracellular signal wouldn't be able to bind to anything because intracellular signals are hydrophobic by character. And if they're hydrophilic now, hydrophilic signals, at least the ones that are involved with whatever signaling they have to do on the surface, bottom line is that that intracellular signal, if it's mutated, it's going to have a really tough time doing anything. It's interesting what you said about it'll be moved to the membrane. I hadn't thought about that. That's interesting. Okay, so I'll move on from that. Okay, so signaling has two general outcomes. It could change the protein or it could change protein levels. And one way that it could change protein levels is through regulation of gene expression. All of these things are obviously very highly regulated. The cell wants to make sure that the signals are doing their things. It doesn't want to make too many signals or too little signals. So I'll give you an example with this. Okay, so some ways that it could change existing proteins is like the glucose example. So let's say, obviously, our blood, we need like a good amount of glucose. We can't have too much glucose and we can't have too little. So what insulin does is insulin binds, it's a hydrophilic molecule. It's a hydrophilic receptor. So is it going to bind intracellularly or extracellularly? It says it on the slide, so that's not a great question. But yeah, insulin, it's a hydrophilic surface receptor, so it's going to find a surface signal. It's going to find a surface receptor. It's going to bind to that, and it's going to tell glucose channels that are inside the cell. It's going to tell them, hey, there's too much glucose in the bloodstream right now. You need to move these glucose channels to the surface. So, like, obviously, once the receptor is bound, those glucose channels are going to migrate towards the surface of the cell, and now glucose has a channel for it to go from the bloodstream to into the cell. Ways that it could change protein levels is by it saying, okay, let's say the cell has detected that it has too much glucose. It could upregulate those glucose transport channels so that it's not in the bloodstream anymore, it's inside the cell. Upregulate the storage vesicles where glucose is stored and used later for energy, or enzymes that break down glucose. And of course, the opposite is true as well. If there's too little glucose in the blood, it would probably downregulate how many glucose channels it would make, because it wants more glucose inside the blood. So it doesn't want these channels promoting glucose to go from the blood into the body. It doesn't want more enzymes that could break up glucose, things to that effect. So does that make sense?
Yeah, I just want to clarify. So like when the glucose, since it's hydrophilic, we know that like they normally wouldn't be able to enter the cell, but like they have those channels that allow them to enter. So when we eat like a sandwich or we have like a bunch of candy, they'll go through that channel, they'll enter our cells, but then when our body detects that we have too much glucose, insulin will bind, and then will it reopen those channels, causing glucose to exit, or what does the binding of insulin do to the glucose?
The binding of insulin to the surface receptor sends a signal down. So once the receptor is bound to the insulin, it communicates information inside the cell. and it tells a cell through, it's probably through like a whole cascade of different proteins. I don't know exactly like all the proteins involved, but it's like a cascade of proteins. So one protein talks to another, that talks to another, that talks to another, that reaches a glucose channel, a glucose membrane channel. And once it gets to the glucose membrane channel, it tells the channel that there's too much glucose outside. It needs to move glucose from the blood into the cell. So that channel is then going to be transported to the surface of the cell. It's going to embed itself into the phospholipid membrane, into the layer, and it's pretty much going to function as a channel for glucose to come inside. Does that answer your question?
Yeah, so glucose is going to be on the outside, and the insulin binding is going to make glucose enter because it's going to open those channels for it.
Yes, insulin is going to open the channels for glucose to go out of the blood into the cell. This is just one example. I don't think like, you're going to have like any like exam questions on like specifically glucose and insulin, any of that. This is just an example of how these cells communicate, or it's an example of how it changes existing proteins, how it moves them around, and how it changes protein levels. So up-regulating stuff, down-regulating things. So any questions about this? Okay. Okay, so our cells constantly need signals. It is always signaling itself to do stuff. Like I said, it is like how cells know how to do everything. It's through signals. They are never receiving like one signal at a time. It's always like a magnitude of signals to do like a bunch of different things, which we'll talk a little bit about later. Our cell's default is pretty much to die. So if our cell stops receiving signals, it's going to undergo programmed cell death. So that's how critically important these signals are. If it's not receiving these signals, it will, it won't be able to communicate what it needs, what it doesn't need. And because of that, it can't really sustain itself that well. Okay, so I have a question for you. Why is it hard to grow cells in vitro? In other words, like on a petri dish or in a test tube or something. Why do you think it's pretty hard to keep these cells alive if you're trying to grow like stem cells, for instance? Do you guys have any ideas?
Yeah, I would just say that because it's in a petri dish, there's no longer those like active and constant survival signals occurring. So without that, it's automatically going to go straight to its default with apoptosis. Whereas like in a cancer cell, it's mutated in a way where those like survival signals are essentially occurring without like actually having to be in the cell, and that's why they can like continue to grow in like a petri dish.
Yeah, exactly. There are so many molecular signals that go into cells to keeping them alive to do whatever they need to do. There's so many signals. And when researchers were first isolating stem cells and they tried to grow them in a petri dish, all of their stem cells were dying. And they're like, well, what's going on? They didn't realize that to maintain these cells alive, they needed a whole bunch of signals. Or they needed like a whole bunch of environmental factors that obviously you don't have in a petri dish, whereas these cells do have inside an organism, inside our bodies, or whatever. So any questions about this? Here's a nice little cartoon about the different things that it could do. So when this cell, for instance, when a cell receives A, B, and C signals, that tells it to survive. But when it receives like these letters, it tells it to grow and divide and, so forth. Okay, any questions so far? Okay, so mutations are obviously pretty bad in most cases. If you damage or you mutate one of the cell signals, that could obviously lead to some pretty bad consequences. A very common example are common cells, cancer cells. They are replicating, they're staying alive, they're doing all these things that they're not supposed to be doing. So I have one more question for you. Which pathway do you think is most likely mutated in the case of cancer cells?
Probably B, C, and D.
Why would you say that?
Because for cancer cells, the cells generally divide uncontrollably. For survival cells, survival. As we mentioned before, the reason why the cells were able to survive in a petri dish, it's because the signal wasn't being sent to them anymore. So the cells just continued to grow there. And then programmed deaths, well, because it's cancer, then that means that the cells won't die; it'll just keep expanding, growing.
Right. So I would totally agree with you. In the case of cancer cells, cancer is just the extreme proliferation of cells where they're not supposed to be growing. So definitely your division pathway is going to be messed up because cells want to, they don't want to make too much of themselves. That's why we have the right amount of cells for our stomach lining or whatever, like the cells that make up our bones. Like it obviously wants a certain amount and wants just enough, but once it reaches that good enough number of cells, it doesn't want to keep making more like heart muscle tissue or something like that. So yeah, division is obviously, the division pathway is definitely going to be pretty messed up because these cells keep dividing when they're not supposed to. The survival signal is also going to be messed up. The programmed death, like apoptosis, these cells, like when our cells do divide too much, it would just be like, okay, bro, like it's time to die. And it would send a signal that would cause its ultimate death. But in the case of cancer cells, because they keep dividing and they keep proliferating, that programmed death pathway is pretty messed up because it can't tell the cell to die. So it will inevitably keep proliferating. So yeah. Okay, any questions about this? Okay. All right, so we talked about some signals. Some signals can have multiple different outcomes, like the picture suggests. So for instance, acetylcholine is a very popular one. Acetylcholine, it's obviously one chemical, it's one signal. It could bind to maybe like one receptor, a receptor that accepts an acetylcholine that's specific to acetylcholine. But depending on where this receptor is, depending on what cell it is, acetylcholine and the receptor could have a very different response. So this just speaks to there very rarely is just one signal that's responsible for like one thing. So, you know, for instance, acetylcholine, as you see here, acetylcholine is not just responsible for like muscle contraction. Acetylcholine is responsible for a whole bunch of different things. And that goes for a lot of different signals, right? So it just speaks to how intricate and how complicated all these signals are. So for instance, in the case of acetylcholine, it's responsible for forcing contraction of heart muscles or secretion of saliva. So there's a whole bunch of different things that could go on with different signals. We'll get a little bit more into this when we start talking about crosstalk, which I'm sure Michelle has a lot to say about that. So do you guys have any questions about this so far? Oh, I closed the chat. I'm sorry. Let me open it real quick.
There are no questions in there.
Okay. Okay, thank you. Amplification, crosstalk. Okay, so signal amplification. We mentioned in class that this was kind of like a game of telephone. Like one person tells someone, I think Keshwar, you were talking about this. I'm sorry, did I pronounce your name right?
Yeah, no, it's pronounced Keshwar.
Keshwar, I'm so sorry. Okay, yeah, you said that in class too, and I tried to like remember, but obviously I failed at that. So I'm sorry, Keshwar.
It's all good. No one ever got my name right on the first try.
It's cool. My friend T Earth, it took everybody a long time for them to learn his name too. So I hear you. Anyway, with signal amplification, so we talked about before how signals are responsible for doing something, right? A signal has to bind to the receptor. That receptor communicates information to a protein, and that protein talks to another protein, and it ultimately like does whatever it wants. However, that's a very time-consuming process. If there's only one signal involved, like if there's only one signal and one receptor and one protein that's supposed to do all of these different things, that's very time inefficient. So what the cell does is this thing called signal amplification, where the ultimate goal out of this is for that one signal to reach that receptor and that receptor to communicate to a bunch of different proteins, like you could see here. So very rarely, right? Very rarely will one signal be responsible for one receptor, one protein that goes to another protein. notice that this signal is activating so many different, in this case, oh, this is CAMP. Okay, so the cyclic AMP is talking to all of its activated PKAs. So that's the general gist of signal amplification. It's to increase the concentration. This is really important. It's to increase the concentration of activated downstream effector proteins, which all downstream effector proteins means is just everything like going in the downstream direction, right? So the cyclic AMP to the activated PK to the tyrosines, whatever. Okay, does that make sense? Does anybody have any questions? Sweet. Okay, here's
receptor proteins.
Oh, sorry. Would you guys like want to watch this video? Is that something you want to do, or just move on from that?
Yeah, I'm fine. I'm fine watching it.
Okay.
Yeah, so 1 minute video I included just because I think some people were a bit confused difference between crosstalk and signal amplification. So I just put it in there, but it's only 1 minute.
Okay, good call. Yeah, I'll let it play. Oh, geez.
Can you make it full screen?
Receptor proteins.
I'm sorry, full screen?
Yeah.
Interact with signal molecules
Can you hear
at the surface
guy?
of the cell.
Okay.
In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified.
Concentrations.
Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP, in turn, will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
Sweet. Okay. So that's the general gist of signal amplification. Do you guys have any questions about that? Okay.
Cool. Okay, so I'm going to chat about crosstalk and then I'll take over the other half of this review. So the next thing we spoke about was crosstalk, and this happens when you have...
I'm sorry.
Okay. This happens when you have components on one pathway that affect another, and the signals are passed between pathways. But basically, this is the way that pathways can communicate with each other, or quote unquote talk. So one protein from one pathway, so if we look at like the growth factor pathway, it's able to chat to the hormone transmitter pathway, so it can block or inhibit proteins from there. So that's why you see all the arrows in that really complex diagram there. And by having the protein from one pathway speaking to another protein from a different pathway, they're able to change the function of another protein in some way. And this is a pretty effective method for integrating a signal. And it works very well when you have multiple components of a pathway. An example that was included in here is when a cell is receiving a death signal to undergo programmed cell death, it should not also be receiving a cell division signal. So one pathway could suppress the other. Does anyone have any questions on crosstalk?
I actually do have a question for you guys. So you could drop it in the chat if you'd like. But do you think crosstalk would speed up the signal transduction? Transducence? I don't know if I'm saying that right. Okay. I'm seeing a couple yeses. Actually, it's actually pretty circumstantial, but I would implore you to think not necessarily. So crosstalk doesn't necessarily speed up the rate that the signal is performed, like from signal to whatever needs to get done, it doesn't necessarily speed that up. What it does, like Michelle says, is it integrates with other pathways. So like the example that Michelle gave, if a cell wants to die and undergo the programmed cell death process, it will be involved with suppressing cell division, but it doesn't necessarily mean that it will speed up the process of cell death. Does that make sense? I remember this was a quiz question when I took this class, and I got that question wrong. So I just want to give you all a fair heads up. Does that make sense? Does anybody have any questions about that?
Could you repeat that one more time, please?
Yeah, so crosstalk isn't really, or it's not necessarily responsible for speeding up the rate that a signal is transmitted. Like it doesn't speed up the signal transductance. What crosstalk is that it integrates itself with other pathways. So one example is, again, like I'll just use the Michelle example. If a signal if a cell wants to undergo programmed cell death, it would receive a signal that would tell it like, okay, I need to die. And that protein, when it's on its way to do whatever it is, so right here, for instance, FADD, when it reaches this, let's say this protein right here may be responsible for suppressing the division pathway, because obviously if something wants to die, it shouldn't be dividing. So it integrates between different signals. It talks to other pathways, but it doesn't speed up the rate that the signal goes through. Does that make sense?
I want to see if I can provide an example for crosstalk that's not related to biology, and just see if it makes sense. So for crosstalk, just picture that you have, like, let's say we have two groups of people, and group A has 7 people and group B has 7 people. And so within the group of people, they're trying to relay a message. So group A's message could be, I don't know, something like, today is Wednesday. So just imagine group A is like a row of 7 people, and the first person tells the second person that it's Wednesday, pass the signal on, and then the second person tells the third person, and so on and so forth, until the last component of the pathway, so the 7th person, receives the message that today is Wednesday. So that's group A. And then if you had group B that had a different message that said Wednesday is 2 days from Friday, then you'd have the same sort of setup where person 1 would talk to person 2, and 2 and 3, and so on. But for crosstalk, it's what would happen is, let's say group A's message is today is Wednesday, but the 4th person down the line has fallen asleep. So the 4th person is unable to tell the 5th person the message that today is Wednesday. So maybe in the group B pathway, or group B group of people, there could be a protein or a person in group B that can tell person number 4 in line of group A to wake up. So that person in group B is interrupting the person or protein that is asleep in group A. So it's kind of cross-talking. If the 2 pathways are interacting with each other, or the 2 groups interacting with each other, in a way that changes the activity of one protein, that person. So the person is able to wake up and relay the message. So I don't know if what I said was complete jargon, or if that made sense, but that's sort of just like an analogy I think of for crosstalk. And then I also have an analogy for signal amplification as well, which is kind of like when you tell somebody like a secret, like if you're gossiping to somebody, I could tell Sebastian that today is October, and then Sebastian can go and tell 2 other TAs that today is October. And then those 2 other TAs tell their 2 friends or 4 friends that today is October. So there's that signal amplification where the message is being amplified from more components down the pathway. So I don't know if that analogy made sense, but that's just the way I try to think of the difference between crosstalk and signal amplification. It makes sense. Somebody said in the chat. Okay, cool. So does anyone have any more questions about that?
Could you speak about the crosstalk? So you said the fourth person in the group B cannot pass message will into the group A to group. The fourth person is like can again like that.
Do you want me to repeat the crosstalk example?
Yes.
Okay. Let's see. So crosstalk is like when you have two separate pathways communicating with each other. So the way I use the analogy is like having two separate groups of people. There's group A and there's group B. And each group has their own individual message or signal that they're trying to send through the pathway. And each person represents a component. So in group A, person one tells person two that today is October. And then each person down the line, person two tells person three, and so on and so forth, until person six tells the last component of the pathway, person number seven, that today is October. So that message is representing the signal that's transduced in group A's pathway. So then group B has a similar setup, but they have a separate message. So they could have a different message. I can't remember what I said was the message, but it's the same idea with person one tells person two, and it goes down the line of people in group B until the last person, number seven, component seven, gets the message, gets a separate message. But I was saying for mine that in group A, if you have um, an individual halfway down the line, so in group A, if you have a group of 7 people and the fourth person is asleep in group A, then person 2 tells person 3 that today is October, but then person 3 cannot communicate to person 4 the message or the signal that today is October because person 4 is a component in the pathway, or a component in the pathway that's asleep. So because of that, um, you need to be able to make sure that the component 4 is awake to hear the message. So what would happen is that group B would sort of interfere with the group A component. So maybe a component from group B would communicate with group A's person 4 and tell them that, hey, you need to wake up, which is kind of like how in the cell signaling, how a death signal can be suppressed by cell survival signal. So another pathway is just getting in the way of a separate pathway, and so that causes a change in protein activity. So that's why when a component from group B tells the person in group A to wake up. So that person is kind of like representing the protein. And so once that protein wakes up, its activity changes, right? So then person 4 can tell the person 5 the message that today's October, and it goes down the line so that the pathway has changed now so that it's active. So I hope that makes sense, but I felt like I explained it better the first time. Did that make sense?
Yeah. Thank you.
Okay, no worries. And this is recorded, so you can hear that analogy as many times as you'd like.
True.
Cool. So we're gonna move on. Okay, so this is just a summary of what Sebastian and I said in the last few slides, but some people like to have a written summary of everything. So signals are small molecules or proteins that bind to intracellular receptors or extracellular receptors. And the same signal binding to receptors on different cells produces different outcomes. And cells receive a whole bunch of signals because if they're not receiving signals, then their default is that they will die. And I already feel like I mentioned this for signal amplification, but it occurs when one signaling molecule is perceived by a receptor, which causes the production of many secondary messengers that can go on to bind to many proteins in the next step of signal transduction cascade, which means a single signaling molecule can cause the activation of many downstream elements, thereby amplifying the signal. So like the example I used for gossip. And then Sebastian also said altering existing protein functions is usually much quicker than gene expression. And then like I described with group A and group B, that the crosstalk refers to interactions between elements of different signaling pathways. So that an element of one pathway or component can affect a component of another pathway. So does anyone have any questions on that? I can't see the chat. Sebastian, are there any questions?
Not yet.
Okay. Cool. So I think we can go to the next slide. So I think Professor Francis also talks about how to shut down a pathway. So fundamentally, to inactivate a pathway, a person should inhibit the effector, which is the very last component of the pathway that's been activated. This is usually the best way that we can shut off a pathway. And this is because the last part of the pathway will usually be the component that activates transcription factors. And these can enter the nucleus and cause transcription of a particular gene. But there are many ways that we can deactivate a signal, which can shut down a pathway also. And some of these methods include receptor-mediated endocytosis. So you basically just rip off the receptor from the cell surface. And then there's also degrading the signaling molecule so that none of the downstream proteins have a chance to do their signaling cascade. And I think I had another example, but I don't know why it's blank there. Oh, okay, I'll just keep going. Then it seems like I described RTKs, but that's coming up in the next few slides. So I'm gonna just disregard the last part there with RTKs. And so yeah, this is just a diagram of signal transduction. Just like I said, you have your components of the pathway, which are like which are in green, and then like your signaling molecule and the receptor. And then eventually all of that signal cascading, the end result of that leads to the last effector of the pathway entering the nucleus, and then you get transcription of a particular gene.
Yeah, I'd just like to add, remember that transcription factors are the ones that are the proteins that are responsible for either promoting or suppressing the transcription of something. So let's say it looks like this one. Let's say this signaling molecule. It's a hydrophilic signal, because obviously if it was hydrophobic, it'd be able to go straight through the bilayer and do its thing. But because it's connecting to a surface receptor, it's a hydrophilic signal. So it attaches to its surface receptor. It has a cascade of proteins doing its thing, relaying information. It's going to find a transcription factor, and it's either going to tell the polymerase to transcribe all of this or suppress transcription. In this case, in this diagram, it looks like it may be like promoting transcription, because obviously we got an mRNA transcript and then its protein's done. So this is just a visual representation of how signals can upregulate or deregulate protein production. Any questions about that?
Cool. So then the next portion of this, I'm going to discuss kinases and RTKs. I just wanted to briefly go over protein kinases. There are multiple different kinases, and kinases are considered proteins, and they're also enzymes. And kinases, their kinase functions are cleaving off phosphate groups from ATP and attaching them to a substrate. And this substrate is normally another protein. And it's important to recognize this because kinases don't just tack on phosphates to things. They're involved in this enzymatic process of cleaving phosphates from ATP molecules and then attaching them to other proteins. Because people tend to think that kinases just phosphorylate things, but they're enzymes. So it's more of a like reaction that's occurring. But in most of our diagrams, you will see that kinases just sort of like have P's on them for phosphates, and then you don't normally see the ATP ADP. So you don't see it like taking off the ATP. It's usually not pictured in the diagram, but just know like in the back of your head that that's happening with kinases. So does anyone have questions on this? I don't know where the words are for the top, but yeah, this is just another picture of kinases. Because a lot of people, I think I saw a misconception that people were confusing the fact that enzymes are proteins, and kinases are proteins too, and enzymes. So it just shows you what kinases do. Cool, we can go to the next one. Okay, so we looked at RTKs, which are receptor tyrosine kinases. These are a family of receptors that are responsible for survival, cell division, differentiation, and insulin signaling. And just like I said before, kinase is an enzyme that catalyzes phosphorylation. And down at the bottom there, I have a picture of RTK, which is part of our RAS MapK pathway, which I will be getting to today also. Does anybody have questions on RTKs? No? Okay. And so generally how RTKs work is that you have your signal that will bind to the RTK, and then that will cause the RTKs to dimerize, which means that they come together, which Sebastian, can you point to the second one? Yeah, that one. Yes, and they dimerize. And then once dimerization occurs, the intracellular tyrosine kinase domains become active due to a conformational shift. Then we have cross-phosphorylation, so each component of the RTK can cross-phosphorylate each other. They cross-phosphorylation of the tyrosines that are within the kinase domain. And then once that occurs, once they are phosphorylated, they are active. So you have an active RTK now, and this can create binding sites for intracellular signaling proteins, which you will definitely see once we get into pathways.
Could you just go back and quickly explain what you mean by conformational shift after they dimerize?
Conformational shift. So for that, I think it's just that when the signal binds to the RTK on the extracellular space, the signal just makes it more likely that the RTK will dimerize. So there's a change in shape, and I think that's what it's talking about. So change in shape, and normally when things bind to other things in cells, it leads to a conformational shift, which leads to change in function. So without the signal being there, that you see on the left, the very first image, when the signal is not there, the tyrosine kinase domains cannot bind together because they haven't undergone that conformational shift. So like shape shifting type of thing, if that makes sense. But once the signal binds, that creates sort of a shape shifting or change in conformation within the RTK part, the receptor that is in the cytosol portion of the cell, which causes them to dimerize, come together. So does that make a bit more sense with that, what I mean by that?
Yeah, so the dimerization, like that coming together, they're undergoing that shift during dimerization, and that's what allows that cross-phosphorylation or autophosphorylation between RTK.
Yeah, because without the signal, you see how on the left without the signal, they're inactive? So they wouldn't be able to cross-phosphorylate each other, even if those two were super close together, because you need the signal, and once the signal binds, it sort of changes shape, the bottom portion of the RTK. And that changing shape is what I mean by conformational shift, which allows both the RTKs to dimerize, and then they can cross-phosphorylate each other with the tyrosine kinase domains. Does that make a bit more sense, Jesse?
Yeah, that does. Thanks.
Awesome. Okay. Any more questions on that? No? Okay. So I'll just keep going. So there are four main pathways we'll be focusing on. They're in eukaryotic cells. The first is the RTK RAS MapK pathway, which I'm not sure if that's even its name, but that's what I call it. And those are used for cell division and cell differentiation. I think that it's also called the growth factor pathway, I want to say. And then the second one we'll be looking at, well, they're not really in any particular order, but it's how I listed them, is the survival pathway. So this involves RTK, PI3 kinase, and AKT, and it's used for cell survival signaling in multicellular organisms. Third one we'll be looking at is JAK-STAT pathway, and it's used, the one we'll be looking at, the example, the receptor is, I believe it's prolactin, which, so the entire pathway will result in milk, production of milk during breastfeeding. So that's an interesting one. And then the last one we're going to look at is the GPCR pathway. I forget what GPCR stands for. But this is the most common receptor in the human genome, and there's 800 versions. Today, I'm only going to talk about the first one, but Friday we'll cover the other three so that we don't bombard you with too much content today. Does anybody have questions? No? Okay. So first one is the RAS MapK pathway. So as usual in these pathways, the receptor tyrosine kinase will begin the show. So your signal comes and it will bind to the receptor. Then you have dimerization of the RTK. And this is what activates the intracellular kinase domains. And then they're able to cross-phosphorylate each other. The cross-phosphorylation of the tyrosines within the bottom portion of the RTK is able to occur. So then the RTK autophosphorylation creates binding sites for intracellular proteins. And does anyone have questions so far? I think that's just similar to what I explained before. But yeah, that's the beginning of it. Okay, so then once that happens, you have adaptor protein or GRAB2, which will bind to the phosphorylated tyrosine. And where the yellow P is, that's actually called an SH2 domain. And an SH2 domain is a domain that makes a binding site for a protein to bind to with a phosphorylated tyrosine. Yeah. So the phosphorylated tyrosine creates a SH2 domain for the adaptive protein to bind to.
Could you go back to the previous slide for a sec?
Yeah. That one?
Yeah. Okay. Sorry. There's something I just had to write down.
That's okay.
All right, I'm good. Thank you.
Cool. So then, sorry, I just feel like I wasn't phrasing things correctly. So you have GRAB2 that will bind to the phosphorylated tyrosine. And then once this occurs, the adapter protein or GRAB2 can recruit the RAS activating protein, which is pictured in light blue, which is the RAS-GEF or SOS. They mean the same thing. And so then your RAS activating protein will exchange GDP for GTP in RAS. And so when RAS moves from a GDP state to GTP state, RAS becomes active when it's bound to GTP. And so basically the RAS activating protein is what changes RAS's conformation. So it has like a lower affinity for GDP, but higher affinity for GTP. So then a GTP is able to diffuse into RAS protein's binding pocket. So then at this point, we have the RAS GTP that becomes active. Does anybody have questions on this portion?
Could you just go back to explain the adapter protein RAS activating protein binding portion? I understand that adapter protein or GRAB2 will bind as soon as the tyrosine's phosphorylated at that SH2 domain. But where is that SH3 domain that she mentions? Is that on the adapter protein that allows RAS-GEF to bind?
I believe so. The SH3 domain is the one that connects the adaptive protein to the RAS activating protein. But I will check it, look it up to make sure. And if I'm incorrect on that, I'll send an email. But yeah, normally SH2 domains are for phosphorylated tyrosines, and the phosphorylated tyrosine creates a binding site for another protein, and that binding site is the SH2 domain for the adapted protein. But I'm gonna actually look into the SH3 one because I do remember she mentioned there's SH3 in the adapted protein, but where it would be, I'm guessing it would be between the dark blue protein and the light blue, because it can't be with the RTK and the GRAP2 because that's SH2. So I'm gonna guess that the SH3 is the other one, but I will look it up after the review to make sure, okay?
Yeah, because I think she mentioned like SH3 also uses GRAB2, so that's why I was a little confused if adapted
Yeah,
protein is GRAB2. So I was
so
just
that
curious.
is true. So SH3 is used by adapted protein as well, but for the connection between the RTK and the adapted protein slash GRAB2, that is a SH2 domain. So that yellow P, that's an SH2. Where exactly the SH3 domain is located, I'm not too sure, but I do remember her saying that adapted protein contains both SH3 and SH2. So I think that SH2 part is important to note and where it's located in this diagram. But I'm gonna make sure that I get an answer to your question about the SH3.
Okay, thanks, Michelle.
Yeah, no problem. Does anybody else have any questions about this portion? No? Okay. I'm gonna move on. Also, I separated the slides by like the different phases, but at the end, there's gonna be one slide that has the entire image so that you guys can practice like how to explain the pathway. But I just like it in smaller chunks to help with the information. Cool. Okay. Sebastian, can you go to the next one?
Yeah, sorry. I just wanted, for the sake of clarity, what Michelle was saying was this part, like what we're talking about. So this next slide is just a continuation of right here. So Michelle explained all the way up until here, the activated RAS protein. And now this is what the activated RAS protein is now doing, in case anybody was wondering about that.
Cool. Okay. So then once your RAS is bound to GTP, it is in its active state. So what does RAS do next? So activated RAS will interact with MapK3, which is the green protein at the top there. And keep in mind that RAS is not a kinase, so it does not phosphorylate MapK3. It just interacts in a way that causes MapK3 to become active. So then once that occurs, you have your MapK3 or MAP kinase, which will phosphorylate MapK2 at two different positions. That's why they're two yellow Ps. Then you will have MapK2 that phosphorylates MapK1. And once you're hearing me say phosphorylation, you should immediately think it becomes active. So MapK3 phosphorylates MapK2, MapK2 becomes active and will phosphorylate MapK1. And MapK1 is phosphorylated by MapK2 at two different positions. Then MapK1 will go on and phosphorylate other proteins, which is what you see with the protein XY, the gray proteins at the bottom, and the transcription regulators there. And so eventually, those transcription regulators at the bottom, I believe those are transcription factors, so those will enter the nucleus and cause change in gene expression. Or you can have the MapK1 can activate proteins X and Y and cause changes in protein activity. Does anybody have questions on this part? No? Okay, we'll keep going. So this slide here is just a review of everything I just said. Some people like to have all the steps written out so that it helps, and other people like the chunks of information delivered in small bits. So this is just a summary of everything I just said. And this slides will be released after the session, so I don't feel the need to read it all again. So does anyone have questions on anything on the slide though? It just explains everything I said.
Could you go back to the last slide really quick?
Back one slide, okay. Did you have a question?
Just want to take note.
Oh,
Thank you.
okay. Sorry. So sorry if I'm going too fast. Feel free to just like yell at me, hey, you're going too fast, and I'll slow down.
Thank you.
Yeah, no problem. Right, you can go to the next one. The next. Thank you. Okay, so this is just what I was saying. I explained the pathway, I broke it down into several slides, but after every pathway, I purposely made sure to put the entire diagram of the pathway as the final slide so that you can go back and you can say, hey, I think I know how to figure out this pathway, or I know what everything in this pathway does. And you can walk through the steps and say, okay, the signal binds, your RTKs dimerize, cross-phosphorylate each other, then this happens and this happens. You can kind of go through and see the sequence of events. And I highly recommend practicing it that way so that you get used to figuring out what normally happens in the pathway. And so then you can figure out problem solving questions like what would happen if there was a mutation in RAS protein, or what would happen if adapter protein couldn't bind to the RTK. Then you could be able to figure out sort of questions like those, which usually she loves to ask on exams or problem sets, ways to think about how the pathway would change or deviate from normal if you manipulated or changed certain things. So yeah, does anyone have questions? No? And I guess that's it. And there also, I wanted to mention that there is a crosstalk video in the slides, which I did not go through for the review session, but I considered it to be helpful because we had one on signal amplification, so I just thought I'd put one on crosstalk. It's about a minute long as well, so it just helps with crosstalk. People need to go back and read that. And so yeah, that's it for today's review session. I'll stick around at the end for questions about anything.
Thank you, guys.